We start our journey as we travel down the esophagus and are emptied into the stomach. As we observe our surroundings, we notice the inner lining of the stomach. A quick scan reveals the surface. It is perforated with microscopic holes, also known as gastric pits. These release enzymes and hydrochloric acid into the stomach to help it digest food. A simulation will help explain the individual cells of the gastric pits. First, we have mucus cells in dark brown. These secrete mucus to protect the stomach from digesting itself. Next are parietal cells distinguished by small flaps. These cells create hydrochloric acid, symbolized by fluorescent green particles, which helps dissolve food. Third are chief cells, shown in light brown. They give off pepsinogen, represented by the purple orb, which transforms into the protein-digesting enzyme pepsin when it reacts with hydrochloric acid. Pepsin is actually one of the only enzymes to work best in an acidic environment. The transformation from pepsinogen to pepsin is symbolized here by the changing of color from purple to blue. Last of all are G cells in dark red. These release a hormone called gastrin, the magenta particles, into our bloodstream, which stimulates the stomach to begin contracting, the release of hydrochloric acid, and the buildup of the mucus layer. It itself is stimulated by nerve messages from the brain, the stretching of the stomach, and the presence of proteins in the stomach. These are some of the main cells in the gastric pits that allow them to function as they do. As we return to our travels, we pass through the pyloric sphincter of the stomach and on into the small intestine. We find ourselves in the duodenum, the section of the small intestine where the most digestion occurs. A small duct empties bile, symbolized in green, from the gallbladder, and digestive juices, in yellow, from the pancreas. The bile emulsifies fats, just as soap emulsifies oil and grease. The digestive juices contain alkaline bicarbonate to neutralize hydrochloric acid from the stomach, and digestive enzymes to break down starch, fats, and proteins. A simulation will help explain how enzymes work. Enzymes are proteins that act as catalysts within organisms by speeding up reactions. This is depicting amylase, a starch-digesting enzyme in light brown. As it meets a starch in gray, the starch attaches to the active site, the part where the reactant is split. Most enzymes need a coenzyme to activate the reaction. In this case, it is a water molecule. After the reaction takes place, the enzyme wanders off to another molecule to repeat the process. It is not permanently damaged through the process, and it can only carry out one chemical reaction for only one set of reactants.